Good evening, everyone. This is Shane Gebauer from BrushyMtnBeeFarm. I'm the general manager here. And tonight, as you all know, we have Maryann Frazier with us. I'd like to thank her for taking some time out of a very busy schedule and spending the evening with us. One of the things that I really appreciate with Maryann and the work that she does, as well as some of the other folks up there at Penn State, is the fact that the research that you're doing, Maryann, is very much applicable to beekeeping. You know, so much we hear about research that's going on that is good information to know, but as beekeepers, we like to have information that we can incorporate into our beekeeping practices. And that's one of the things that I've always admired from you, is that much of what you do is applicable to day-to-day beekeeping. And that's really, I think, what a lot of people want. As I do these presentations, it seems that that's what people are asking for. So one, I just want to express appreciation for the work that you do. In particular, this topic of pesticides in the beehives is something that's of a lot of concern to a lot of people. I think it has a tremendous impact in, and I don't want to steal your thunder or go into anything too much here, but it seems to me that it's got potential to have a tremendous impact on colony health. And so I'd like to thank you for taking some time. I know you're very busy traveling around, meeting with organizations. You're a fantastic speaker and sought after for good reason. And so I really do appreciate you taking some time to be with us this evening. And before I turn it over to you completely, I would just like to say one thing, and it seems appropriate given that you are affiliated with a research institution and that a lot of the money that funds research nowadays is in the form of grants. I have to just take a moment and do a shameless plug here for a product that's been brought out onto the market this year. It's put out by Bee Lover's Garden, and it's a calendar. It's a 2010 calendar that a bunch of people have put a tremendous amount of work in. And the woman that sort of spearheaded this, her name is Mel Hughes, and she's actually on with us this evening. And she's worked with a lot of artists in the North Carolina area, in the Asheville area, to pull together some beautiful, beautiful illustrations of plants that are benefited by pollinators, but in particular honeybees. And the reason why I'm taking the time to mention this is that a portion of the sales from that calendar are going to fund honeybee research. We've got this product on our website. It's also available through Bee Lover's Garden. If you Google that, it'll bring you right there. And it's an excellent opportunity for holiday gifts. It's also an excellent opportunity for beekeeping associations to raise a little money. They can buy it in volume, get it for a little bit lesser price, and then sell it to coworkers, colleagues, family, friends, whoever. And make a little money for their bee association too. So given that we're talking about some research here that's been that you've worked on, Maryann, it seemed appropriate to put a shameless plug in for that product. She's done a lot of hard work on it, and all in the spirit of trying to help the honeybees. She's actually not a beekeeper herself, but she feels compelled to help us in the industry. So thank you, Mel. I know you're listening, and we appreciate that, and hopefully we'll get you some sales as a result of this shameless plug. I normally don't plug products specifically, but this seems sort of like an exceptional situation. Now, one more thing before I turn it over. For the benefit of Maryann and also myself, I have some poll questions. Those of you that have tuned in the past, you're familiar with these. I'm going to go ahead and launch these poll questions. This just sort of helps guide the conversation and target the information at a level that everyone can benefit from. So here's the first one. Whoops, I clicked the wrong button there. I'm sorry. Let me try that. Let me try that again. Here's the first one. How are you controlling Varroa? Just choose the one that's most appropriate. Go ahead and select one. Are you using hard chemicals, things like Checkmite or Apistan? Soft chemicals, things like Apivar Life, Apigard, Mitaway 2. Non-chemical IPM methods, drone trapping, varroa screens, things such as that, or a combination of these factors above. If you can go ahead and just select one, we've got 51% in, 54, come on now. I'll go ahead and close the poll in 5, 4, 3, 2, 1. I'm going to close the poll. Last minute votes. Go ahead. All right, we're closing that poll. Let me share the results. In terms of hard chemicals, we've got 5%. Maryann, can you see these results?
I cannot see.
Okay, so it's, as a presenter, unfortunately, only us viewers can see them. That doesn't help you much. But let me just read off the results here, and maybe you can quickly scratch them down. Hard chemicals, we've got about 5% of the folks. The soft chemicals, we've got 35%. Non-chemical IPM, 47%, and a combination of these factors, we've got 12%. Let me close those, and we've got one more poll that we're interested in here. Let me go ahead and launch this. Do you recycle your brood comb? In other words, do you sort of have a culling regiment? Yes, about every 3 to 5 years you have a complete rotation of your brood comb. Every 5 to 8 years or so you've got your rotation as needed. So a frame breaks, or you happen to notice one frame that's just loaded up with wax moth or maybe tons of drone, and you feel that you want to just get rid of that one frame, or no, you haven't done any brood comb culling. I'm going to go ahead and close this poll in 5, 4, 3, 2, 1, and I'm going to close it. Last minute votes coming on in. All right, closed, and I'll share this. And Maryann, for your benefit, I'll read off these results again. On a 3 to 5 year cycle, we've got about 47%. On a 5 to 8 year cycle, we've got 5%. And then as needed, 24, and no, also 24%. So there are the results of the polls. Thank you for taking the time to answer those questions. And with that, Maryann, I've hogged enough of your presentation time here. I apologize for that, but once again, I do thank you for taking some time to be here with us tonight. I really do appreciate it, and it's all yours.
Well, thank you, Shane, very much. First of all, for putting this together, I really applaud you and Brushy Mountain for the effort to do this very innovative technique. These webinars are something that I've been really interested in participating in and learning the technology, and so you really gave me the opportunity to be able to do that. So I'm really very, very thankful to have this opportunity, and particularly to be sitting here very comfortably in my office and be able to share this information with so many people. It's really a fantastic opportunity. So I'm really, really thrilled to be here, to be a part of it. Thank you for asking me. Thank you for that very kind introduction. And I do want to mention that I myself have one of Mel's calendars, and I'm torn between whether I want to give it away as a gift or keep it for myself. So I may have to actually indulge in buying a few more of those calendars.
That was the answer she was looking for, right there.
Well, I am fairly certain I'll have one in my office and enjoy it throughout the year, so I'm sure other people will as well. It's a beautiful, beautiful piece of artwork, and nice information, of course, about bees and beekeeping as well. Beautiful present. So, okay, on to the topic at hand, the issue of pesticides in our beehives. As a lot of you probably know, maybe some of you do not know, that we have been involved here at Penn State quite heavily in the work to try to understand what's happening with our declining honeybee populations and in relationship to CCD. And so we have different teams of people working on different aspects of this, and we have a team that's working on the issue of pesticides. And so I'm going to share with you sort of where we are in that effort and all that we feel like we need to do in order to try to understand the role of pesticides in our beehive. So I'm going to start here. That's interesting. Okay, so insects, bees, we know, I don't have to tell a lot of you who are beekeepers tuning in here that bees are built for pollination. They're the insects that have this close relationship with plants and are really designed for collecting pollen, using pollen, and in the process pollinating our plants, our agricultural crops, as well as a lot of non-agricultural crops as well. So we know that there are, we have a lot of bees in this country. We have 3,500 species of bees, but of course honeybees are very, very important to us in terms of pollination of agricultural crops, mainly because we know and understand them and can manage them quite effectively. We are able to move them where we need them, when we need them for pollination. So they're very mobile. They're available in large numbers in the early spring. If we're talking bumblebees, yeah, bumblebees are out there in the apple orchards in the Northeast, but there are only queen bumblebees out there, and so that doesn't make for very many bumblebees. And in apple orchards, we need a lot of bees early on to get the job done. They're well studied. We understand that we need to move the bees into the crop. For instance, in apples at the time of 10% king bloom to attract the bees to the crop. So they're well studied. We understand the relationship between the bees and the plants, and they're mobile. They have this floral consistency where once we get them on the crop, they're going to be faithful to that crop. So all these things about honeybees make them the ideal pollinator for our large agricultural crops. Part of the reason why this has come about is because our agriculture today is, of course, in the form of large monocultures. Many of our crops consist of large blocks of a single crop, where there's a fair amount of pesticides used to control the pest insects, and there are not a lot of other alternative plants available, weeds, i.e. weeds, that compete with the crop, that are important for our pollinators during the season whenever the crop is not in bloom. So these, U.S. large monocultures, use of pesticides, use of herbicides to eliminate competing plants, means that the use of mobile pollinators, bring them in during the time that the crop needs to be pollinated and be able to get them out whenever the crop needs to be protected from pests via use of pesticides, makes honeybees really our ideal pollinator for so many crops. So they're very, very important to not only beekeepers, but to growers of many crops and to most American consumers who want a varied diet, an interesting diet. So we know, many of us, that the value of honeybees pollination is quite high. 15 billion. We've seen figures as high as 19 billion, and this is just of our agricultural crops. We don't even have a value on the non-agricultural crops, on the food for wildlife, for instance, and the diversity of plants in our many ecosystems, and the contribution that honeybees make to the fertilization and reproduction of those plants, and the food, that nuts and berries and so forth, that feed our birds and wildlife. So very important to us. I'm not telling a lot of you, I'm not telling anything new. But honeybees, as well as our other native pollinators, are certainly in trouble, and I think a lot of people are aware of this to a great extent because of the phenomena known as CCD. So pollinators in trouble, and I always try to point to two very important publications, I think, that demonstrate the breadth and width of this trouble that pollinators are in. This is a status a publication on the status of pollinators in North America that was produced by the National Academies of Science. This is a body that's asked to comment on important scientific topics and research scientific topics to provide to policymakers and so forth about the status of a particular issue. And this study was 4 years in the making, and it was in publication at the time that CCD was first manifested itself. And so what this publication is saying, in a nutshell, is that pollinators in North America are in decline. Not only honeybees, but all of our pollinator species are in trouble. And another publication that I think is also very important and telling is this science article by researchers that looked at pollinators. These are not generalist pollinators, but more specific pollinators, pollinators that are specialized on certain plants in Great Britain and in the Netherlands. And what they found was not only are the particular bee species in trouble, but the plants are also in decline that are dependent on these pollinators for their pollination. So the take-home message here is that it's not just honeybees that are in trouble, it's all pollinators, and it's not confined to North America. We see this decline in many parts of the world. It's a little easier to see in North America and in Europe, I think, because we have a better handle on documenting things in these places. So CCD, in my to my way of thinking, is the manifestation of an overall decline of pollinators, the latest manifestation, if you will, of the overall decline of pollinators. It's very real, it's very unique, and it's very devastating. It has been very devastating, and I think is going to continue to be for many beekeepers. What we see in this colony collapse disorder is that these colonies, as you can see here, were, I don't know if you can see my pointer or not as I go here, but these are framed. You can see that, Jim?
Yep.
Okay. So these are frames. These were taken by Jeff Pettis, a beekeeper in Georgia, and we see that these colonies, these frames, have very nice, especially this comb right here, frame of solid, nice solid frame of brood. This is sort of capped brood, what we would like to see in our colonies in general as we look at them in the height of the brood rearing season. Nice brood patterns, solid brood patterns, very few uncapped cells here. Here we see the beginnings of the bees emerging. So these were the first laid eggs and the oldest brood emerging. And on the outer edges we see the more mature brood, capped brood that will emerge shortly. The point here is that there's a lot of brood here. This colony was obviously quite strong and healthy to produce this amount of capped brood, and yet it almost looks like somebody's shaken the bees off of these frames. And what we have are basically empty frames with the queen present and what are probably just the newly emerged bees from all that capped brood there. So basically the loss of the adult population. So this is a really nice illustration of what we mean when we talk about CCD. So this happens very quickly over sometimes a week, sometimes maybe a week or 2 weeks, and in some cases within a few days. The beekeeper goes there at one point, and by the end of the week he goes back to check on those colonies, and we see the loss of the adult bee population. So this is a mystery. We still to this day do not understand what has caused these adult bees to leave this colony. It's very unusual to see adult bees leave their brood and the resources, the nectar and pollen that would be in this colony. So very, very unusual situation. Whenever this first happened, the Apiary Inspectors of America commissioned a very, very quick survey of beekeepers in the United States, and this is actually published in the American Bee Journal, this survey, and they found that in non-CCD operations, losses were a quarter of the bees were lost, which is high in and of itself, but in CCD operations, beekeepers lost on average 45% of their, or overall 45% of their colonies were lost, not on average, but 45% of the colonies. In some cases, beekeepers lost as much as 90% of their bees in a particular operation. So overall that year, we saw a loss of a third of the colonies in the United States, and that has not really changed much over the last 2 winters, 2008 and this past winter, 2008-2009, we saw nearly a third of the bees lost in each of those years. So this third of the honeybee the colonies in the United States being lost each year is very devastating and is not sustainable long term. Yes, beekeepers are working to replace their losses, and they're doing it effectively. That's why we don't see no bees at this point in these large operations, because they constantly are feeding and trying to build up what's left of the bees and split and replace the lost colonies, which they do effectively, but there's a tremendous amount of labor and resources going into making up these losses. And so the beekeepers are wearing down financially and physically and emotionally, as well as the bees. So we're equally as concerned about the bees and the beekeepers. So, what is responsible for this decline in pollinators? In some cases, we know what over time what has been responsible for some of the sharp declines we've seen. Varroa mites, for instance, tracheal mites, resistance to chemicals used to control varroa mites. These things have all led to sharp declines in honeybee populations. In other cases, we've seen things that happen to the bees from year to year that time to time that we haven't been able to explain. Fall dwindle disease, for instance, spring dwindle. There have been things that big losses from time to time that we haven't been able to explain.
Maryann,
sure.
I'm sorry, but the percentages that you showed in that previous slide. Now, the date you gave a year there, 2007, 2008. These are over the winter, right? These are sort of winter losses. This isn't sort of from January 1 through December losses, or are they?
Right. Now this is a survey that's done in the spring of the previous, so 2007, that survey would have been done like in March of 2007, and it was taking into account the losses right over that winter of 2006, 2007. That's correct. And the same with 2008. Those are sort of the losses during that period, during that sort of basically over the fall, winter, and early spring. Yes.
Okay. Yeah. Are there any numbers that sort of look at whether the sort of growing season or active season losses are any different than they have been in the past?
I don't think so. I'm not aware. I don't know if you are of any surveys that are done sort of the industry at large.
Right. I'm not aware of anything, but it's not uncommon to have a couple colonies go queenless, or we had some here in this area that absconded because there was a dearth and they were desperate looking for something. So you always sort of have those sorts of issues, and I'm just wondering if maybe along with the increased percentage, along with the increased loss in wintertime, if there's also an increased loss in sort of the growing season as well, to sort of only emphasize the point that we're losing a heck of a lot of colonies.
Right. Yeah, I think it would be maybe worthwhile to look at that. I just think that because we see such dramatic loss of colonies during this period of time, the late fall, the winter, and the early spring, that sort of has been the focus, especially because it was kind of driven by the whole CCD thing.
Sure, yeah.
That's sort of when we saw those losses. There have been some studies, like the study that Jeff Pettis and Dennis Van Engelsdorp were looking at on migratory, where they followed certain migratory beekeeping operations, and they've documented at each crop, for instance, the number of colonies lost, at each stalk throughout the season. So I think there have been some kind of focused studies, but I don't know of any sort of big overall studies that have looked at monitored losses throughout the year.
Okay, thanks.
Sure. So of course, it's been difficult to try to narrow down what it is that we should really be looking at in terms of why these losses are occurring. But the things that have kind of come to the cream that has risen to the top of the pile here, if you will, are things like the pathogens. Are we seeing a newly introduced pathogen, or potentially a newly evolved pathogen that has become more deadly to the bees? Are there pesticides, beekeeper-applied pesticides and farmer-applied pesticides, agricultural-type pesticides, that are responsible? Or are there environmental stresses like poor nutrition and genetics that are contributing? And at this point, I will just say that we think all of these things, and including the one missing there, is that we know is responsible for losses, is varroa mite. And so these things certainly are things that we think are contributing combined to reduce pollinator populations, honeybee populations. The issue with CCD also seems to be the potential that there's a combination of things going on here, and maybe not even always the same combination of things that lead to this collapse of the bees known as CCD. But I'm going to kind of focus on the pesticide issue because that's the one that we're working on here, our team is working on. So I'm going to spend a little bit of time here talking specifically about the pesticides. But I do want to stress right off the bat that we don't think pesticides alone are the cause of CCD, but we do think they're an important factor, certainly in pollinator decline and possibly, probably in CCD. So having said that, let me go into a little more detail about that. So honeybees can be exposed to pesticides in lots of ways, in particular whenever they're taken off to a pollination situation where they're visiting a crop that they're pollinating, and although the growers are quite careful in most cases about not exposing their pollinators that they're paying for to pesticides, it does happen. Also, bees will visit crops that we don't necessarily think that they're going to visit or want them to visit, like corn, for instance. We wouldn't expect bees to be pollinating corn. It's a grass, it's wind pollinated, but bees do visit and will collect pollen from corn. And so those people who are designing pesticides for corn, protection of corn, aren't thinking about pollinators. And yet we see pesticides, or see bees certainly collecting corn pollen and potentially exposed to pesticides that are used on corn. And lastly, of course, we as beekeepers are using pesticides in our colony, although from our survey we can see that not a whole lot of you are using hard chemicals like coumaphos and fluvalinate in your colonies for control of varroa mites. And we are very concerned about that exposure as well, and I'll explain more about why. So this is the team that's working on this this issue here at Penn State. This is my husband, Jim Frazier, Chris Mullen, Jim is a physiologist, Chris is a toxicologist, and both of these individuals are we're very thankful to have them on this team. There's no way we could do this work without a toxicologist particularly, and a physiologist. They've been really important to the effort. And the others, Daniel Schnell, Daniel's a graduate student working with us, Sarah Ashcraft, and another graduate student, Wan Yi Zin, and two other undergraduates who are technicians in the lab are very important to us. In addition, I always mention that we do have very strong women working in our group. We test them all ahead of time to make sure they can handle hives, and the women are quite strong or very smart. And we also have Craig Sella, who works with us quite often, especially when we have heavy-duty jobs to do, like weighing colonies and/or installing a lot of packages, that kind of thing. We're very thankful for Craig's participation. So our team collaborates with other teams that are involved in some of which are involved directly in the CCD, the pollinator decline effort, and other teams that we have thankfully brought into the work. So here, this is USDA and Dennis Van Engelsdorf, so Jeff and Dennis, who have done a lot of the sample collection, that's them and their teams, a lot of the sample collection for the CCD versus non-CCD bees, the migratory study, and so we were very thankful that they're out there in the field doing all the sample collection, and we basically get the samples back here at the lab and are able to then process them and evaluate them and so forth. So our collaboration with other members of the CCD working team has been really important. These two people up here, Roger Simons and his technician Maria, are at the Gastonia lab in North Carolina, and they have been really important to us in terms of and the data analysis. It's been really tough to find a lab that's capable of doing the kind of analysis that we wanted to have done, and they have the equipment and the knowledge to be able to do that. In addition, Roger Simons is a beekeeper, and that has been really helpful. He understands bees and beekeeping, understands the language that we speak, is very sympathetic to the problems that we're having, and so he's been really important to this effort, and we're very thankful for that lab and their work. So to date, we have about 900 samples that we have analyzed, and these have come from the CCD study, an apple orchard study, which I'll tell you a little bit more about later, the migratory beekeeping study, and beekeepers submitted samples. We do have a cost-sharing program that is funded by the PAM, Preservation Apis mellifera, and Foundation for the Preservation of Honeybees, that allows beekeepers to submit samples. We pay half the cost through the grant, and the beekeeper pays the cost. So that's been really, really helpful, and that information is confidential but does go into our database. So we have the data, not connected with the beekeeper's name in the database, but the data, and then the beekeeper, of course, gets the results from the analysis.
And Maryann, maybe you're going to get to this, but what's the cost of that, and how do people go about submitting a sample? Because I'm sure that in just about 2 more seconds, that question's going to be coming streaming into us.
Absolutely. So the cost of the analysis right now, and this will go up at the end of the year, currently the cost is $205. We pay $105, and the beekeeper pays $100. In order to participate in the cost-sharing program, the beekeeper has to send me an email and tell me that they're interested, and then I send them the protocol on what they need to do, how they need to package the sample and send it to me, and with the check and the little information sheet on why they're sending the sample. Either because they expect that the bees are exposed to pesticide, or they just want to check to make sure their pollen or their beeswax or their honey or their bees are without pesticides. Okay. So and I'll give you my email address at the end of this, so you can send me an email if you're interested in participating in the cost sharing program.
Can they send them in from Canada?
Unfortunately, because the funding sources, our 2 funding sources have asked, we've had a number of requests from other countries to have samples analyzed, and the funders have asked that the samples come from U.S. beekeepers. Okay. However, having said that, beekeepers from anywhere in the world can send samples if they're willing to pay the $205 to have the analysis done. That can certainly happen. Okay. Thank you. Sure. So the screening of the samples, we screen for 171 different pesticides. We have to develop a screen concerning what we're going to look for. This analysis isn't just you put in the sample and you get out what pesticides are there. You have to have standards, and you have to know what it is you're looking for in order to be able to look for it. So we have a screen that looks for 171 pesticides, and we can detect at the part per billion level. So this is very, very minute levels of pesticides. We can, or Roger's lab, I should say, can detect, their equipment can detect at levels as low as 1 to 5 parts per billion. We've looked at wax, bee bread, trapped pollen, adult bees, brood, and royal jelly, and very small amounts of royal jelly and brood. For the most part, it's been wax, pollen, and bees. And we've looked at samples from a lot of large migratory beekeeping operations, but also a number of small and even organic operations as well. And I'm sorry, I meant to update this, but we're up to 23 different states that we've looked at samples from, at least from 23 different states. This is the list of pesticides that we have found. Of the 171 that we've looked for, we've found this number. This is 121 pesticides. So this is surprising to us to find this diversity of chemicals in our colonies. But what is more of a concern is that we have found very few samples that have lacked any detections. Almost every sample we've looked at, probably less than a half a dozen, maybe at the most a dozen, have had no detections of the 900 that we've looked at. Of these 121 pesticides, metabolites are the breakdown products of the pesticides, and sometimes they can be as toxic or more toxic than the parent compound is. So that's important to keep in mind. Of these pesticides, we have found all classes of pesticides. These are broad categories of pesticides: pyrethroids, organophosphates, carbamates. Neonicotinoids is one that there's been a lot of interest and concern about, a new class of pesticide. But growth regulators, organochlorines, chlorinated cyclodienes, herbicides, fungicides, the miticides are represented here. Of these, 14 of these different pesticides are systemic. Systemic means that the plant, that's applied to the plant, either the seed or in the water that's irrigating or fertigating the plants as young plants, or even applied to the plant itself, and the plant sucks up that pesticide and translocates it throughout the, I shouldn't say it really sucks it up, but it is absorbed into the tissues of the plant and is translocated throughout the tissues. And of course, our question is, well, what is the chance that it's being that it's being expressed in the pollen and nectar as well, that pesticide, and the bees are picking it up and bringing it back to the hive. And so this is the big concern about systemic pesticides. And the neonicotinoids are all systemics. Some of these other pesticides are also systemic, but this whole class of neonicotinoids is systemic. So that's one of the reasons why there's a lot of concern about the neonicotinoids. They are also toxic to honeybees. There is variability in their toxicity, but a lot of them are quite toxic to honeybees. This is one of our big concerns here, is that it's not just one or two pesticides that we find in a pollen sample or in a wax sample, but on average, we are seeing 6 pesticides in a pollen sample, on average. Up to 31 pesticides in a single pollen sample and 39 in a single wax sample. So what this means is there's a lot of diversity of chemicals in these samples. And you think about pollen, pollen is the food that's being fed to the developing bees, and young nurse bees are consuming a lot of pollen in order to activate their brood food glands. So we're really concerned about the potential for interactions with these consumption of these different chemicals. If you think about what the doctor asks you when you go to the go to a doctor for a visit or go to the pharmacist, one of the first things they're going to ask you is, what other drugs are you taking? Because they're concerned about prescribing you a drug that potentially could interact with the ones you're already taking. Well, we think the same thing is highly possible and likely in a honeybee colony when we're seeing multiple pesticides of different classes. So this is in part sort of where we are. I'm not sure why that's doing that. There we go. Okay. So just quickly, I don't want to spend a lot of time and get bogged down here a little bit on these details, but this is an interesting slide because it talks. We looked at some samples of trapped pollen as well as bee bread that were from the same colony, and what we found, which was not surprising, was that trapped pollen had higher levels of the chemicals outside the hive, and bee bread had higher levels of the chemicals that we find inside the hive. But what was surprising was that we found any coumaphosphine fluvalinate in trapped pollen. If you think about, a trap on outside the colony where the bees are out there foraging and they come in and they're dropping their pollen before it even gets into the hive, how is it that this coumaphosphine fluvalinate is getting into that pollen? Yes, we can see in the beehive that the pollen could be absorbing, and we think this is true when it's packed in the cells. There is this coumaphosphine fluvalinate in the wax, and it's being translocated from the wax into the fatty pollen, into the pollen which has a high lipid content. But how is it getting in the pollen that doesn't even come into the hive? We think that the bees themselves are contaminated when they're out there collecting the pollen. They are actually incorporating these pesticides even before the pollen gets into the beehive with these chemicals that we see so so much of in the colony. So that's a concern. I just wanted to quickly go over, okay, so that we have this large list of pesticides. You know, how many pesticides are there in large enough quantities or in diversity to make a difference? What does this all mean? You know, how important is it? And so there's a lot of things to take into consideration. So here's a little piece of the data that we have and sort of the ways that we look at this data. So here on the left-hand side, we see some of the chemicals that we find that we're particularly concerned about. So one of the first things we are concerned about, and whoops, sorry, think about, back up there, is the LD50 here. How lethal, how toxic are these chemicals? Chemicals have a varying degrees of toxicity to honeybees, and we measure this through the LD50. This is the lethal dose, so that's going to kill, the dose that's going to kill 50% of the population. And so we call that the LD50, or the lethal dose at 50%. That will kill 50% of the population. So you see there's quite a range here of LD50s. One of the reasons why we're concerned about things like the neonicotinoids, this is the class of insecticide, imidacloprid is one of these, and the LD50 is quite low. The lower the number here, the more toxic, the less it takes to kill 50% of the population, so the more toxic it is. Where if you look at things like coumaphos, we see 46,000 parts per billion is the LD50. It's quite high, so it's not so toxic to the bees. So that's one of the first things we need to look at. And then, of course, we need to look at how often is this material actually detected. Well, there's a big variation there. In the neonicotinoids, the ones that we're maybe concerned about being systemic, because they are systemic and they're toxic to the bees, we don't see them in our samples very often. In the samples that we've looked at, 836, we've only picked up neonicotinoids 14 times, or imidacloprid, I should say, not neonicotinoids, but imidacloprid, that particular chemical, only 14 times. These are the metabolites of that pesticide. However, coumaphos and fluvalinate we see in a very high percentage of the samples. In this case, this is pollen, and so in this case we see coumaphos and fluvalinate in 75% and 84% of the samples respectively. So in a lot of the samples we're seeing coumaphos and fluvalinate. So how often they're detected is important, the frequency of detection, and then next the big thing is how much of the material we actually seeing. So we look at the mean and we look at the low and high values to understand how much material, how much of these pesticides is actually showing up. And in some cases we can get pesticides that are nearing or above the LD50. And so here you can see a pesticide, this particular one, sorry, losing my arrow here, this particular situation where the highest value is well above the LD50 here. And so that would be a case where we would be really concerned and expect to see some lethality, some toxicity of that particular pesticide.
Maryann, I'm wondering, with the imidacloprid, its total detections are relatively low, but I'm wondering about maybe some of the metabolites that you mentioned earlier. You know, and I'm thinking specifically about some work that Jeff Pettis, I think it was, did, where they took, I'm trying to remember this correctly, where they took colonies, they actually fed these colonies known doses of imidacloprid, and they then tested the, well, it was the bees, I guess, maybe not the wax, but they tested the bees that were fed the imidacloprid bee bread, and they couldn't find it. But then they inoculated colonies with nosema spores, and what do you know, the ones that were fed imidacloprid had much, much higher rates of nosema infection than the controls, despite the fact that they couldn't find imidacloprid in the colony.
Yeah, I think this is a big concern, and it's certainly, we are not taking the neonicotinoids and imidacloprid off the table, because we know that in some cases the bees can metabolize the pesticide very quickly. So in other words, it goes through their systems quickly and we don't pick it up. That doesn't mean that it hasn't had an impact on the bees. So even though we see very little of it, the same is true with amitraz. We don't see the amitraz parent compound at all because the bees metabolize it. What we do see are the metabolites. We see the breakdown products. We have two metabolites of imidacloprid. You can see them here, the olefin and the 5-hydroxy. But there are other metabolites of imidacloprid that we do not have the standards for. So we cannot look for them. And this is a concern, that two concerns. One is that that some pesticides are metabolized so quickly that we don't pick them up in our screen, and secondly, that the metabolites the things that they break down into, we don't have in our screen, so we don't see the metabolites. So that is a concern, and it is a shortcoming of this situation of just looking at the screen data of the pesticide data. It doesn't tell the whole story by any means.
Okay, and the reason why you don't have a full array of the metabolites is?
If you ask our toxicologist, he'll say you have to know what the metabolites are in order to request them from the pesticide company. And if you don't know what the metabolites are and they may know, they don't freely offer them. We don't know exactly what they are. We don't know what to ask for. We don't get them. So
I see.
There's a little struggle there. It would be very helpful if we had all the breakdown products and had them in our screen of certainly of the neonicotinoids, but we don't, and that has been a frustration for us.
Okay, and one last question: what's the LOD column there? That you've got?
Yeah, the LOD is the load of detection, and that is the lowest level at which it can be detected. So this 5 right here is 5 parts per billion. That's the lowest level that we'll see it at. In coumaphos, we can see as low as 1 part per billion. In the metacloprid, we can see as low as 2 parts per billion. But in the metabolites, we can only see as low as 25 parts per billion. So it's the lowest level at which we can see the pesticide. There are some screens that have even lower levels of detections, below a part per billion, but they can't have as broad a screen. So we've sacrificed low levels of detection because we don't think they're that important in order to have a very broad screen.
Okay.
Does that make sense?
Yes.
Okay. So the last thing I just want to point out on this chart is that there are some other things that are coming up quite frequently, like this chlorthalonil and this clopyrifos. One is an organophosphate and the other is a fungicide. And one of the things we've learned in the lab and also has been documented by other researchers is that fungicides can synergize with certain classes of pesticides, insecticides, excuse me, to be much more toxic than they are independently. So here you can see that chlorthalonil is not very toxic at all. The LD50 is 111,000. Actually, it's even more than that. Parts per billion. Very not non-toxic. Over a million parts per billion. So not toxic at all. But when you combine these pesticides, these fungicides, some of them particular classes of fungicides, with insecticides, they synergize so that they become more toxic than those are individually, or even additively. And this is we think is a big problem. There has been one publication that's documented that these things can become, fungicides can become 1,000-fold, or even the insecticides can become 1,000 times more toxic than they are when they're combined together with fungicides, the fungicide and the insecticide. So I'm not sure I explained that exactly right, but the idea is that when you combine the fungicides with insecticides, particular classes of fungicides and insecticides, they synergize to can synergize to become much more toxic.
So is there any way to sort of calculate an LD50 for these two particular compounds that you've got circled here? Obviously, it would, well, I'm assuming it might vary based on concentrations of each, but are there any sort of studies that look at that to say, okay, you look at the one and it's got a LD50 of 1220, well, does that drop down to below the 280 that we're seeing for the imidacloprid?
Yeah, that's one of the things that we're working on, and I'm going to show you a little bit of data about that here in a minute.
Great.
That is definitely one of the big questions that we're asking, and it's very complicated because not only are we looking, sometimes at 2 chemicals, but sometimes you're looking at 3 or 4 chemicals. And then what can happen, which is even very strange, and what we're seeing in our lab toxicity studies, is that sometimes they can cancel each other out. So 2 things might be more toxic. You add a third, and it becomes less toxic. So it becomes an antagonist.
Whoa, okay. Yeah.
And so yes, it can become very, very. And when you're talking about an average of 6 pesticides per pollen sample, it can become very complicated and very difficult to tease apart. But let me go on here and show you some of this data with the synergy that might help explain some of this. So, but in general, I just want to say that we are seeing pesticides from in-hive miticides in wax and also agricultural pesticides. Think of wax as a big sponge. It basically picks up and holds. These pesticides are lipophilic, most of them. So they dissolve in a lipid. They want to dissolve in a lipid, in a fat. Wax is very high in lipid content, so they do dissolve and are held in. I shouldn't say dissolve necessarily, but they are held in wax. We think from the data that we have looked at, movement of pesticides from wax into pollen. And both agricultural type pesticides as well as miticides from in-hive use. We have looked at foundation and found that foundation across the board is contaminated with coumaphos and fluvalinate. So even whenever you put new foundation in the colony, you potentially are introducing or will have a baseline of miticides and potentially a few of the agricultural chemicals as well. We're very concerned about brood food. Here are these young larvae. They're in contact with the wax itself, and then with the brood food, what is the chance that some of this pesticide is moving from the wax into the brood food? And then, of course, there's pollen that is in later older larvae, is applied directly to the brood food. What about the chances of this contaminated pollen being added to the brood food and the larvae consuming that? So this is a big area that we're looking at right now, and others as well. Jamie Ellis in Florida, as what's happening to these larvae when they're exposed to this high levels of pesticide and diversity of pesticide in these colonies. So the question no longer is, are there pesticides present in the beehive? But that was sort of the first hurdle to get over. And are they there in levels high enough and a diversity of chemicals enough to be even concerned about? And the answer to that is absolutely yes. The pesticides are there. They're there in enough diversity and high enough levels to be concerned about. So now the question becomes is, what kind of impact are they actually having on the hives themselves? So we're looking at this in a number of different ways. The toxic effect of the pesticides, so multiple pesticides, that combination additive effect of those pesticides, what's the toxic effect? And then the synergistic effect, particularly for certain insecticides and fungicides. Behavioral effects. We know that the chemicals that we see, chemical class that we see most often and at highest levels are the pyrethroids. And we know that these pyrethroids interfere with memory and learning. And we're really worried about that potential for these pesticides to interfere with bees' ability to learn the location of their nest and be able to forage successfully. So our graduate student, Daniel Schmel, has been working on this learning and retention. Physiological effects, like on longevity, does it shorten the lives of the bees if they're exposed to one or more pesticides? And the potential that they're interfering with the immune system. And we're doing that in combination with Diana Cox Foster, and then this work on larval development and what happens to those larvae if they make it to adulthood. Is their behavior and physiology normal if they've been exposed to pesticides? And so that's another area that we're looking at in detail. So this is just a couple of slides that show the kind of ways that we do this. Here's Sarah. She's done 53 of these different toxicity. Actually, she's done a lot more than that now, but she's done a lot of these toxicity in-lab toxicity studies. I tell you, she's really getting tired of. She calls it killing bees, and that's exactly what she's doing here. Is she looks at chemicals singly or in combination, and of course always with controls to determine if there's what the toxic effect is, and also if we see behavioral changes in these bees. Of course, in cages, that's a little bit of a tall order, but certainly the toxic effect of the pesticides can be measured easily in these kinds of cage studies. So here's one of the examples of what we were talking about earlier, where she's looking at two different materials. This is this is another big hurdle that we face, looking at the technical material versus the formulated material. Technical material is the chemical by itself. The formula is how the growers would buy it. And we're really concerned that there are things, additives, adjuvants that are added to these pesticides that may in and of themselves be toxic to the bees. So in this case, we are using a formulated material, and we're looking here at a Sale, which is a neonicotinoid, and Nova, which is a fungicide at 3 different doses, and then she's looking at them in combination at 3 different doses. And what you can see here is that the Assail and Nova, each by itself, over this 24-hour period, there's no mortality here at all. Whenever she combines these together, we see a very, very, this is the synergistic effect. We see mortality very early on, even at the second dose. Here we're at the LD50. This is a probit which basically calculates the LD50 for you, which would be 5 here. We should have had a 5 on here, but we're above the 50%, so we're above the LD50 here at the second dose of this combination of pesticides. So this is an example of a nice example of a synergistic effect. And we've done a lot of combinations like this to try to understand how these pesticides are working. So this is the 24 hours. This is 48 hours where we start to see some mortality in the Assail and Nova, but we never reach the LD50 within the 48 hours. Yet again, this we see the synergy. This is sort of the same figure that you saw earlier, but we just in comparison here to show you that we do see mortality from these other things over time, but it is quite low compared to the combination. The other issue that we're really looking at in detail, I'm going to just check the time how we do. It's 7. Are we okay, Shane, time-wise?
We're fine. I mean, other than feeling a little low right now with all this pesticide talk, we're doing just fine with time. Don't you worry about that.
All right, all right. The memory and learning is an interesting assay, and it's one that's certainly not been developed by us. It's used by a lot of labs around the world to ask questions about honeybee memory and learning. This is an associative learning assay where the bees are presented with an odor and with a reward, a sugar reward. So here the bee's been puffed with a little bit of an odor and given this reward. And in 1 or 2 trials, the bee learns to associate the odor with the reward, and you can present the odor without the reward, and the bee will extend its mouth parts. So we can treat bees with pesticides, feed them pesticides, known quantities and doses of pesticides, and we can have controls that we have not done that with, and then test how long it takes them to associate, for instance, the reward with the odor, and then how long they retain that memory. And we definitely have seen pesticides, and this is not new information, but neonicotinoids are one group of insecticides where we can see a definite interference with learning and memory in honeybees. And we're not the only ones to be able to demonstrate that in the lab. Other people have done that as well. And certainly there are other pesticides that can also interfere with this ability of bees to learn and remember. And of course in honeybees this is incredibly important. They have to be able to memorize the location of their nest so that when they go out and forage, long distances away from the hive, they can find their way back to the nest. So incredibly important, and for sure pesticides can interfere with this capability in honeybees. Oops. Okay, longevity is another thing that we look at, pretty straightforward. We can take bees from certain situations, like this particular case where bees were on pumpkins, and we take newly emerged bees, one-day-old bees, as they're emerging, we let them emerge actually for 24 hours and then separate them into cages, and we determine how long those bees live. We can actually do that. Of course, we always do this with control bees as well, bees that haven't been in a pesticide situation, and we can determine how long those bees have live in those cages. So here's an example of a case where we were looking at bees that were from a control colonies, 3 different control colonies, and then 3 different colonies where the bees were on pumpkins, and the year before hay was on that land hay was grown on that land, and a second case where pumpkins were being foraged on, and the year before corn was grown on that land. And this is just one way that we were looking at it. We also tested, of course, to see if certain pesticides were there, but if we see an effect from pesticides that we know were applied to corn, we were concerned about that the year after whenever there's a bee-pollinated crop there. And you can see that we do see a difference here. This is about right for bees that are in cages that are not fed pollen but only sucrose syrup. To live for about 22, 23, 24 days is about average. And here we see the pumpkins and the hay is not too different from that, and yet we see a lower level on average for the pumpkins that were previously, bees that foraged on pumpkins that was previously corn ground. But again, this is one of our struggles with honeybees, is there's a lot of variability here. And even in this case, the colony that lived the longest, the bees that lived the longest, were one from this group. And so again, when we look at the averages here and do a statistical analysis, we don't see, with this small sample size, a statistical difference. So there's no significant difference between these 3 treatments.
Is that being replicated to see if you can, yes, with a larger sample size? It seems like only 3 samples per treatment is pretty small.
Exactly right. That's exactly right. So what we did this year, and I'm sorry I don't have the data here for you, is we have a study, which I'm going to tell you about, in apples, where we took bees to apple orchards, and we did this with 27 different colonies. So I will mention a little bit about that here in a minute, but I don't have the data here to show you. But we did see a difference in longevity of the bees that went to the apples and were treated or were exposed to pesticides, and those that were our controls. We did see differences, a big difference in the longevity of those bees. So yes, not in this particular situation, but certainly in other other research projects, we are doing this same assay chain.
And just out of curiosity, are you also are you moving the controls just to replicate the movement into a pollination center? You are very thorough.
Yeah, we're trying to be. We're trying to be, but it's amazing. It took us 3 years to get that study right, and I'm going to tell you a little bit about that apple study in a minute. But what I wanted to just point out here is this variability. Here we see this variability that exists in colonies, and Shane is getting at is that it takes a lot of colonies to actually get at the story, to see a pattern that is significant. And we also think that, I told you about a lot of things that we're doing with bees individually. Even this longevity work is basically with individual bees. The toxicity work is with individual bees. The learning and memory retention is with individual bees. And yes, we can do a lot of things and see the demise of individual bees, but when we try to move to colony level work, it's much, much more difficult. And we think that bees have the ability to withstand as a colony a lot more than they have the ability to withstand as individuals. So if you think about a bee overwintering, one little bee by itself cannot possibly survive the winter, but the colony can. And we think that they have mechanisms that allow them to do that. And we think that's true for things like diseases and also things like pesticides, that the colony as a whole has mechanisms for dealing with problems that individuals don't. So the same thing is true, you know, whenever we go out and do an experiment, we try to, you know, buy packages from the same producer with the same, you know, sister queens, and we introduce them into these colonies. Within a week, we see a tremendous change in, you know, some colonies have 10 pounds of bees, and some colonies have a pound of bees. And so, you know, this is part of the struggle that we face as researchers doing this kind of work. In addition, varroa mites complicate the questions at the colony level. Nosema management, whenever we're looking at our own management as well as management by a number of different beekeepers, if we're looking across you know, a number of different operations to try to get an answers. So I guess I'm going to kind of finish up here with this apple orchard study and go into a little more detail about that. These are 3 orchards where we've done this study for the past 3 years, where we know the pesticide history on these orchards for many years, for 8 or more years. And the growers are very cooperative, and they tell us exactly what they apply and when. In addition, within each of these orchards, there's 2 treatment blocks. There's a reduced risk block and a conventional block. And the reduced risk block, they actually is where they use the neonicotinoids, and the conventional is where they would spray routinely. And what the researcher Dave Bittinger is trying to do here is look at whether or not they're seeing an increase in the number of beneficial insects in these reduced risk blocks. In the control, so we move the bees into the orchards during bloom. We move our controls not to an orchard, but to a place where there are some apple trees, and then we move them all back to a home apiary after the bloom. And then we monitor those colonies with we do a lot of different analyses on these. We know that the pesticides that they're being exposed to, there are 2 orchards that have high levels of exposure, 1 level that has an intermediate level of exposure, and then we have our controls, which really do, from the past 3 years, we've been able to document that they have little to no pesticide exposure. We monitor individual bees for longevity, for glandular development, for gene expression in terms of their toxicity genes turned on and off. And we look at colony level responses like the growth, the health, the survival of colonies, the disease levels, the mite status, and the reproduction in those colonies. So we're trying to monitor all of this. When we bring the bees back to the home apiary, we put pollen traps on them so that they cannot collect additional pollen, but have to feed on the pollen that they've collected in the orchard. And so this forces us to know that they have, and we, of course, we take samples of that pollen, and we know exactly what the pesticide load is in those colonies. In addition, we use osmia because osmia don't forage as far. We have seen our bees move from their blocks to the blocks that they weren't supposed to move to. But with the osmia, they stay pretty close to, you know, right in the blocks where they're supposed to. So we get a better handle on sort of, we collect the pollen from them and analyze the pollen to know exactly what the pollen in that particular block has pesticide levels in there. So that's been helpful to use these solitary bees as well. And I will just say we're analyzing the data from 3 years of that study now, and it's pretty interesting what we've learned from that. But it is amazing how resilient these bees are in terms of being able to put up with the levels of pesticides that we've seen. We are working closely with the Environmental Protection Agency. This is Jeff Pettis, and I gave a workshop to the EPA on honeybee management because they're very concerned about the issues around pesticides and honeybees, pollinators. We're working with them on registration requirements and reevaluation of some products, labeling to improve pollinator protection, and incident reports for beekeepers to provide EPA with incidents or information when they have a pesticide kill. And this is just the summary slide very quickly. I'm going to tell you sort of where, what we would summarize here is that it's been very surprising to find as many pesticides and in the quantities that we've seen in pollen. We are seeing the pesticides in the highest levels are certainly the miticides, particularly from fluvalinate, specifically fluvalinate and coumaphos, but there are well over 100 other pesticides and metabolites that we see. Pyrethroids are the dominant class of pesticides. No individual chemical is likely to explain CCD from the data that we've been able to generate. Systemic and other fungicides occur at levels that synergize with particularly pyrethroids, organophosphates, and neonicotinoids. So again, the idea that these fungicides can synergize with these classes of insecticides to be more toxic. And we're very concerned about the role that pesticides might play in relationship to things like viral diseases. Do pesticides interfere with the immune system so things like IAPV can be really toxic, or really virulent, I should say, to the colonies? And lastly, pesticides involved in pollinator decline are most likely, the issue of concern is that there's combinations of these pesticides in bee food, and they are most likely synergistic actions of multiple pesticides that are acting at the sublethal level on particularly behavior and physiology. And so this is where we think the pesticides are really doing their damage. And there's been a lot of public concern, as a lot of you well know, and a public interest, which has, I think, worked to our advantage, in our favor. And we've had a lot of funding sources that have been very, very important to this work. And I actually, this is an older slide, neglected to mention here, Wyman's Blueberry Growers, which just provided us with a grant of $50,000. And when growers were providing money for this kind of work, it's very, on one hand, satisfying and on one hand scary, because they do recognize the potential dangers here of losing their very important honeybee pollinators. So we're very thankful for all these funding sources, and we couldn't do this work without this very important sources of funding. And this is the Merrick website, where we are trying to keep up with the information on CCD. However, I'll just mention that we have lost, unfortunately, our secretary, who has been very helpful at doing this, and so the Merrick website is not as up to date as we'd like for it to be. But I will stop there and be happy to answer any questions that, Shane, you might have from the viewers.
Oh, you've got quite a list here. So take a drink of water, get comfy, and there's some really great questions that have come in. First off, we'll start with sort of some of the general ones. I'm wondering about the distribution of CCD at this point, and also I'm wondering too if you have a sense of just how reliable the sort of reporting of colonies lost from CCD really is. I mean, my experience is that a lot of times people call in, their colonies died off, and they're not really sure why. You know, there's sometimes we just can't put a finger on it. You know, as you've mentioned numerous times, it's complex. So I guess I'm wondering how accurate is the reporting, and what do you think the distribution of CCD really is?
Yeah. It is a good question, and I think it is a little bit difficult to put a really good numbers to, in part because in areas of the country, I think it is widespread. First of all, I will say that I think it is widespread across the country. The reason why I think it's hard to put our really hard and fast figures to it is because the criteria used for understanding CCD is the loss of adult bees in the presence of a lot of brood. And that is easy to document in places where brood rearing is going on, like in the South, for instance, or in California, where there's constant brood production and bees have brood. But what happens in December in Canada, you know, or January in Canada, when we see collapse of a lot of colonies and there's no brood present? That has been a little bit difficult, and people have argued whether or not that is really CCD or not, because it doesn't fit the strict definition of CCD, this loss of adult bees in the presence of a lot of brood and resources. So I think that what we're seeing is a lot of loss of colonies, and I can tell you there's a lot of reports coming in right now from Texas and from the West of operations that are going downhill fast, and we're seeing a loss of a lot of colonies. Whether or not that's CCD, you know, I can't say. I don't know, because one of the things that's not happening now is there was a team out there kind of monitoring what was going on, actually helping operations determine whether or not they actually were experiencing CCD, and we're not seeing that right now. We don't really have a good, solid definition that works in every environment at every time of the year to explain CCD, and that has been a bit of a problem.
Okay. We've got a couple questions here about some of the organic acids that are used for mite control, like oxalic acid and Mitaway 2, which is a formic acid product. Do you see any sort of do you see these things in the wax or bee bread at proportions that are sort of not typical of a healthy hive?
Yeah, those things are not in our screen. We
Okay.
don't have those soft chemicals in our pesticide screen. And so there have been some studies that have shown that some of these things are potentially problematic in honeybee colonies. My feeling is that at this point they are the best options for varroa mite control that we have, even though there are some negative aspects of using some of the materials, the essential oils and or acids. Particularly, we want to stick to those that are registered, like the formic acid, Mitaway 2 pads. But the advantage, I mean, varroa mite is very, very alive and well, and continuing to cause a lot of problems for us. And I think one of the best things we can do for ourselves as beekeepers is keep our varroa mite populations under control. And those things, even though they're not perfect and have their negative aspects, are most likely ways of safest ways of controlling varroa mites at this point.
Okay. As you might expect, there's a couple questions here that are expressing concern about the quality and safety of the honey. We've talked a lot about the wax. We've talked a lot about pollen and bee bread. But I don't recall you mentioning anything about the honey. Are these sorts of pesticides showing up in the honey as well?
Yeah, that's a great question. And whenever this material started to be shared, whenever we started to actually learn what we were, the USDA lab, the lab that we are having the analysis done, is the lab that's responsible for monitoring pesticides in the food supply. And so when they saw the levels of pesticides that we were seeing in pollen and wax, they got a little alarmed, and so they initiated an effort to look at the honey supply and monitor, add it as one of their commodities for a year, and monitor pesticide levels in honey. And what they found was that there was very, very little pesticide in honey. For the most part, it was coumaphos and fluvalinate at levels below allowable residue level. There are tolerances set for coumaphos and fluvalinate, and nearly all of the samples that had coumaphos and fluvalinate had it at levels that were below tolerances set for those pesticides. So this is not surprising, since pesticides, most of them are lipophilic and are much more likely to move into a lipid as opposed to a liquid, an aqueous solution that like honey is. So, excuse me, thankfully we don't see pesticides in the honey. We see them in the wax and we see them in pollen. And I feel fairly comfortable saying that the honey supply is safe and free of pesticides.
Okay. I'm wondering if you could just do me a favor. We've got a couple questions here about some of the pesticides that you detected, that big sort of horrifying list that you showed us. If maybe you could just have that up in the background while we continue with the questions, if it's not too much trouble just to go back to that. There's a really good sort of insightful question that I want to make sure we get to here. This is regarding some of the fungicides that you found in your samples. We know that when the bees make their bee bread, they pack the pollen in, they mix a little honey, but it also goes through a little bit of a fermentation process. And so this person's curious to know if you're seeing any sort of impact from those fungicides that are affecting that sort of preparation of their food, affecting the yeast that's causing that little bit of fermentation.
Right. Yeah, that's a very good question and a hot topic right now. Diana Samataro and Gloria DeGrande Hoffman at the USDA lab in Tucson are really pushing an effort to look at this very question. Not only pollen, but also bee guts themselves. What are there are microorganisms there in the bee gut at work, and what are the chances that the fungicides in particular are interfering with the transformation of pollen to bee bread, and or interfering with the microflora in the bee gut. So they have a huge push on at that lab to look at that question, and I think that's a really critical one. We don't know the answer to it, but we are really concerned about that very thing when we see the levels of fungicides that we're seeing in pollen. What is its ability to interfere with the transformation of pollen into bee bread? So I think a good question, one that we don't have the answer to, but certainly that lab is dedicated right now to working on that.
Okay. Here's a pretty good question, I think, and one that we talked a little bit about in preparation of this talk this evening, Maryann, but we didn't really discuss just yet. So this is a good segue. The topic of comb culling. This person's wondering if you see a pesticide level buildup in wax that's a year old, 2 years old, 3 years old, etc., or have you done that sort of chronology to see that sort of trend, or if there is a trend?
Yeah, no, we have not looked over time at buildup of pesticides in wax. However, we certainly have analyzed comb from a lot of what look to be quite old brood combs, and in some of those we see some of the highest levels of pesticides in some of the what look to be the oldest combs. But we also know things like the half-life of fluvalinate is 5 years, and so once if you get a buildup and they even stop using the material, you're only going to reduce that accumulation by half over a 5-year period. So there is no doubt in our minds that there is not only an accumulation of these things and a buildup over time, but we think a real recycling within the colony itself. For instance, we can take cappings wax and have cappings wax analyzed and find coumaphos and fluvalinate in some cases, surprisingly high levels in the cappings wax, which we wouldn't have expected. We would have thought that in cappings wax that there would be very little to no coumaphos and fluvalinate, and yet in some cases we're finding it. And so we think that the bees are recycling the wax. Again, it's on their bodies. They're eating the pollen that's in the, and possibly even, eating some wax and chewing on the wax and moving it around, and in so doing, consuming and or having that pesticide on their bodies and transferring it all over and even secreting potentially new wax that has the coumaphos and fluvalinate in it. So I'm not sure that answers the question, but there's no doubt in our minds that there is a buildup over time, and how long it's going to take to get that out of our wax supply is a huge, an important question, and I think it is going to take a long time before we really eliminate that or minimize it in our wax supply.
There's a question regarding some work that you did looking at foundation, and were the chemicals found in new foundation or old as well, or what sort of things?
This is all new foundation.
Brand new foundation, okay.
New foundation, and we bought the foundation from the manufacturers of all the large beekeeping supply companies, and we even looked at foundation, plastic foundation, and washed the wax off of that plastic foundation, and found fairly high levels of coumaphos and fluvalinate in all the samples of wax foundation that we had analyzed.
So there's sort of some questions of desperation. One person saying, so are we just expected to lose about a third of our colonies each year? What things can a hobbyist beekeeper or smaller beekeeper do to try and mitigate the impacts of these pesticides? I mean, even if you're trying to be as organic as possible, you're going to be your bees will be bringing these things back to the colony. So what sort of things can we do? One person's looking to also put on their boxing gloves and try and change current laws and fight the chemical companies. So really, honestly, Maryann, I'm feeling a little gloomy right now.
Yeah,
give us some sunshine here. What can we do to keep our bees alive with all this information that you've just shared with us?
Yeah, I wish I could tell you right off the bat sort of what, I wish I had a good answer for you. Mm-hmm. I will tell you that there is a lot of work being done on behalf of our honeybees. There is a group of beekeepers. These are mostly large migratory beekeepers from both the honey producers and from the Federation, who are working together in a panel with EPA and NRDC and USDA to address this problem. And they have been very active and have been, of course, are very concerned and are trying very hard to change policy. And we're working closely with them to feed them with the information, the research, and help them understand the research and data to be able to do that. And I really have a lot of faith that those people are going to make some real difference there. That advisory board is going to really have a serious impact. Beekeepers themselves, I think one of the things that is helpful, although very hard, is to try, and I know the commercial beekeepers are doing this, whenever they move their bees to a crop that they know the bees are being exposed to certain levels of pesticides, they try to rest their bees now in between by taking them to a place for honey production where they're not going to be exposed to a lot of agricultural type crops. And I do think that we need to think about how do we, here at Penn State, we analyze our bees at our apiary that's local here on campus, and it's not that there's no pesticide being used here. Certainly there is, but we really do see a minimal amount of pesticides being brought into those colonies. So I think where you put your bees is what I'm trying to say is important. And if you can try to keep them away from agricultural crops where there are pesticides being applied on a regular basis, I think that's beneficial. And also, you know, homeowners use a lot of pesticides, unfortunately. And so I think one of the things that beekeepers can do, because people are so concerned about bees, is to make them understand that whenever they use pesticides, their neighbors, their friends and neighbors, if you're a beekeeper that keeps bees in your backyard, that the use of pesticides around the home, for instance, for dandelion control, as well, that's an herbicide that they would be eliminating a foraging source for the bees. They want to get rid of dandelions and clover in their yard. Well, you know, that's one of the or two of the plants that bees are really dependent on for nectar and pollen. Mm-hmm. And the use of insecticides in and around their homes and gardens can impact the pollinator populations in their area and the honeybees. So I think we as beekeepers really need to start helping the population at large understand that it's not just agriculture, but it's homeowners as well that are using pesticides and are potentially impacting pollinators. We did one little study where we put the homeowner product of imidacloprid onto crabapple trees in a place where they hadn't actually received any other of this kind of pesticide before. And it was very, very surprising that homeowners can actually apply pesticides, some pesticides, at much higher levels per plant than can an orchard or they can in an agricultural situation. And so I really think that we need to help homeowners understand that they are part of the problem. And we really try to encourage people. A lot of people ask me, what can we do to help the bees and the beekeepers? I encourage them to think about their own use of pesticides. I encourage them to buy honey from local beekeepers to support their local beekeepers. It's amazing how many people want to become beekeepers now, and so that is one of the good things about this. And so the more people that become beekeepers or support beekeepers, the more people I think there are that are going to want to help protect the pollinators and think about, you know, our overall use of pesticides and how we what we're, you know, basically doing to the environment. And I don't think it's just bees. You know, I do wonder what is the potential that that pesticides are having impacts on other organisms like bats that are declining and frogs that are declining. And there is some evidence, particularly in the frog and in salmon populations, that pesticides are a piece of the puzzle when it comes to those population declines. So I think as beekeepers we can really, you know, our goal is to get this information out to you and to understand the impact of pesticides, and the beekeepers can really carry the banner for our efforts to think about pesticides and use pesticides more carefully and more wisely. I think we've really moved into a scenario where we're using pesticides liberally, and I think that it's not good.
Yeah, I guess I would tend to agree with that. I'd also I'd be interested to know if I'm assuming you haven't, but it would be interesting to see if you segment out your data based on operation size. You know, I'm making an assumption here that a lot of the people tuning into this are not running thousands of colonies. I don't see any of the commercial beekeepers on this webinar right now. You know, I'm suspecting that most of the folks here tend to be the smaller operations and that are in their backyard and perhaps not in these agricultural systems that may be subjected to a lot of pesticide application. And so I'm wondering if you would see a different sort of result if you looked at that sort of beekeeper as opposed to the big commercial guy. Now, I know you're getting samples from a lot of different people, but you've also noted that on the Penn State campus that you're not seeing a whole lot of pesticides showing up relative to some of the others. So I'm wondering if that might be an interesting trend. You know, I'm also seeing here that some people are having a little bit better survival in town. I'm also seeing this person obviously is from Canada, that there's regulation now going through Canada I'm guessing more at a local level, that's banning sort of the cosmetic use of pesticides. I'm assuming by that they're sort of saying the dandelion treatment, so to speak. The dandelion's your friend. It's the bee's friend too.
Yeah, right.
And they're, but I'm wondering, there's a question here about, this is a little off topic, but genetically modified organisms, if you're aware of any work that that may be impacting the colonies from those organisms, from the genetically modified organisms.
Yeah, there's been that keeps coming up. That keeps coming up as a potential topic to look at, and it's not something that is off the table, certainly, in terms of potential contributing factors. But the genetically modified crops, particularly corn, and the insertion of the Bt gene, we know that that toxin is not toxic to honeybees itself. So it's a toxin that's inserted that's particularly toxic to Lepidoptera, so things that moths and butterflies and the immatures of those, like corn earworms, for instance. It's effective against those things. So the toxin itself, and there's been a lot of work. Galen Dively, there's a paper by him, Galen Dively, on the Merrick website that talks about, we asked him to write something about GM crops and what the potential is based on his research and his experience for being toxic to honeybees. And basically he is saying that from the research that's been done, feeding of even GM corn pollen to bees has not been detrimental. But we wonder, are GM, we know now that when we insert a gene into a plant, that other changes can occur. And is it possible that something is happening to those plants that makes the pollen less nutritious, for instance, just as an example? And that hasn't been, that kind of thing has not been looked at. So again, it's the idea of sublethal kinds of effects, or not outright toxicity, but other kinds of impacts on behavior and physiology. And that, I think, is unanswered. That we don't know the answer to that. The outright toxic effect of the GM crops to pollinators is unlikely. But again, alternative type, other kinds of impacts are a possibility.
Okay, just one more point of curiosity here. The person that's from Canada said the entire province of Ontario is in 2011 apparently banning cosmetic pesticides. So that's kudos to the Canadians. That's admirable, I think. I'd like to just make one point here, and maybe you've got some comment on this, Maryann. The importance of nutrition. I was really quite surprised. I think it was in Reno this past year at the ABF meeting, the American Beekeeping Federation meeting, that they really seemed to harp on the value of nutrition, which is fairly obvious to many of us. But the fact that it's actually being sort of promoted as a means to hive health, shocker there, huh? That it can actually offset a lot of these. Perhaps the impacts of the pesticides, of disease. I'm also thinking back to that work that Jeff was doing with nosema, that I think he found that once he fed them, that a lot of the nosema sort of cleared up with the added nutrition. And it makes good sense. I mean, we can see it in our own lives, that when we eat right and we've got a good balanced diet, we tend to be healthier than when we eat poorly. And so, that's something that we shouldn't overlook, that can offset, I think, maybe not necessarily the pesticides. Maybe you've got a comment there, Maryann, but certainly the value of nutrition in our colonies cannot be overlooked, that it does have the ability to offset a lot of, at least, diseases and things that otherwise might compromise the colony. But do you see anything like that on the pesticide front, the value of nutrition there? Absolutely.
I mean, honeybees are generalist pollinators, meaning that they not only pollinate a variety of different crops, but that's because they do that because they need a diversity of pollen for healthy brood rearing and healthy adults. So they need a nice diversity of healthy pollen, and whenever we eliminate that diversity, we see this. For instance, when we take bees to crops like blueberries or cranberries, where the pollen, they only get one kind of pollen, and that pollen is not a high quality pollen, and on top of that, they're being exposed to pesticides in that pollen. So that is like a double whammy for the bees. Mm-hmm. And the beekeepers have definitely, one of the things that they have been doing that they think, and I agree, has been useful, is that they're feeding pollen substitutes all the time, pollen supplements all the time now. That's what they do. They are constantly trying to combat this CCD problem and declining health of their colonies, whether it's CCD or not, by supplementing the nutrition in those colonies. And they will tell you that, and I think it's true, that it has been helpful. It has been one of the things that has helped to either minimize the effect of the pesticides and or help the bees combat diseases. We don't know exactly the mechanism by which it's working, but it makes, as you said, perfect sense. If if you're a healthier organism, you're going to be able to fend off diseases and pathogens and potentially toxins like pesticides. Whereas if you're not healthy, you're going to be more susceptible to those things.
Mm-hmm. We've gone on now for about an hour and 45 minutes, which is a little bit longer than I anticipated. So I do thank you for staying with us, Maryann, and being gracious enough to answer these questions. I really do appreciate it, and I know you're busy. This has been absolutely fantastic. To hear this information sort of opens our eyes, I think, a little bit to the bigger picture of beekeeping and how sort of the what's going on around us affects the colonies that are in our backyards or down the road from us. So I really do appreciate that.
You're welcome, Shane.
There is one question here that people are interested about if they want to send samples. Can they? How do they do that? You mentioned getting these your email address or emailing you for the information. How do they go about that?
Right. Yeah. The best way to do it is just to email me at mfrazier. That's MFRAZIER at psu.edu, and tell me you're interested in the cost sharing program, and I will send you the instructions on how to do that. And it's quite a lengthy list of instructions depending on whether or not you're sending a pollen sample or a wax sample or a bee sample, and that's why I don't want to try to do it over the internet here. And we also have a list of questions that we ask the beekeeper to fill out. So just a little questionnaire, 3 or 4 questions about the sample and why they're sending it. And so if you are interested, I'll send you the information about the cost sharing program and the instructions immediately when you send me. If you put, I would ask that you put in the subject line cost sharing program, pesticide cost sharing program, so I recognize that immediately, and what I do is I see those and I immediately shoot off the instructions. We have about $10,000 in that fund, and we will analyze samples until that money is gone. And unless we get more money, we only can analyze samples until as long as we have funds. So I really encourage people who are selling pollen, for instance, and or comb honey, to think about getting their wax and or pollen analyzed. And we're happy to do that, and I'm happy to send you the instructions. Please feel free to just email me, and I will email you the information back immediately.
All right. Thank you very much. Again, I really do appreciate it. And also, if you've missed their email address, I know that if you just type in her name into Google, you'll find her and her contact information. So, Maryann, again, thank you very much for taking the time and meeting with us this evening. and maybe we'll have you back in a year or so to go over some of that apple orchard study that you're doing.
I'd be happy to do that, Shane. Thank you very much. I've enjoyed it very much.
All right. Thank you very much. And everyone, you all have a wonderful evening. Good night.